Welcome to the final segment of Chapter 9. We're going to be discussing fermentation, crosstalk, and regulation of these pathways. Now, crosstalk might not be a familiar term, and you probably won't find it in the textbook, but it's often a term used for when products of a pathway or components within a pathway start working in other pathways. So a lot of the products from separate pathways. Remember, we don't only eat sugar, we eat other things as well. So those things are broken down during different pathways, but then their products can be used, let's say, in at some point in the cellular respiration pathway to continue that process and produce ATP. So let's kind of do a quick reminder of what glycolysis is all about. And the main purpose of this is because when we're talking about fermentation, fermentation uses glycolysis as well. That's the first step in fermentation, both processes of fermentation, as well as anaerobic respiration and aerobic respiration. So it's a common pathway. So remember, glycolysis is the splitting of sugar, right? So let's say the splitting of glucose. Remember, glucose is a 6-carbon sugar. It's split into 2 pyruvates. Remember, during glycolysis, there's an energy investment phase and an energy payoff phase, and glycolysis occurs whether or not oxygen is present. Now, this is key because fermentation will be the remaining part of the process after glycolysis if oxygen is not present. So, during glycolysis, just a quick reminder, remember there are 2 NADHs made and 2 ATPs made. Okay, so for each cycle of glycolysis, that is all. Remember, glycolysis occurs in the cytoplasm. And then if oxygen is present, then the products of glycolysis, or at least the pyruvate and the NADH, will eventually enter the mitochondrion. Now, the pyruvate becomes oxidized and then goes through the citric acid cycle, and then more NADH and FADH2 are produced, which will carry those electrons that they're holding to oxidative phosphorylation. So we've already kind of discussed that in the previous segments. Now, the point here is to remind you that glycolysis, again, is a common pathway among the starting point among all these pathways that we're talking about. And again, it occurs in the cytoplasm. So when oxygen is absent, fermentation will occur in the cytoplasm. Again, the inputs of glycolysis in terms of your book, we are discussing the breakdown of glucose. Okay, so glucose is broken down into 2 3-carbon molecules called pyruvate. And remember, there's 2 ATP and 2 NADHs that are produced as outputs along with pyruvate during the process of glycolysis. And again, this is the first step in aerobic respiration as well as anaerobic respiration. And of course, fermentation falls under that category. Now let's look at Okay, so fermentation and anaerobic respiration allow cells to produce ATP when oxygen is absent. Okay, so during situations where, let's say, you're running out of low oxygen, your muscles will start to undergo fermentation, specifically lactic acid fermentation. And that's where lactic acid will build up in your muscles and often cause muscle cramping and pain. Looking at the one process of fermentation. Now, this looks a little bit busy. This is alcohol fermentation. This is not what happens in your muscles, but other organisms can undergo alcohol fermentation. Looking at this, remember, we have glycolysis. Remember, this is the same glycolysis as what we discussed for cellular respiration. The same things occur, right? So glucose is an input. There's 2 phases. There's the energy investment phase and the energy payoff phase. And ultimately, you end up with a net amount of 2 extra ATP. Remember that analogy with the money, right? So you've made some money. You've made 2 ATP. And that's really the point of fermentation, is to try to produce energy or potential energy, right, in the form of ATP, so that you can keep doing work in the absence of oxygen or the organisms. So organisms that undergo alcohol fermentation are often microorganisms, and of course yeast is one of those. That's how alcoholic beverages are made. So remember, at the end of glycolysis, we have 2 pyruvates which are produced. Now, the two pyruvates are going to undergo decarboxylation. And once they're decarboxylated, remember there's two of them, so two carbon dioxides are released, and then this produces acetaldehyde. Now, acetaldehyde is going to receive the electrons from NADH. So here, during alcohol fermentation, the NADH does not go to the mitochondria and drop off those electrons at the electron transport chain. It's going to drop the electrons off at acetaldehyde. Once acetaldehyde receives those electrons, NAD plus is made, right? So the NADH has lost its electrons. Now it's NAD plus. It's the oxidized form. So basically, NAD plus and NADH are going to switch between the oxidized form and the reduced form during this process, every step of alcohol fermentation. And the result is ethanol. So you can see ethanol right here. So that produces alcohol. Now, during this process, some carbon dioxide is going to be released. Now, there's lactic acid fermentation. This is only one step. So this is like what I described, what happens in your muscles when oxygen is very low. So again, glycolysis is the same thing as every other time, right? So two ATPs are made. Now, remember, two pyruvates are the products of glycolysis. In this case, the pyruvate is going to accept the electrons from NADH, which will then become oxidized, right? So it's going to switch still between oxidized form and the reduced form. And the pyruvate immediately becomes lactate. Okay, so lactate is lactic acid, and that's an acid, so that's going to lower the pH wherever this is building up. So this could build up in your muscles, and lower pH makes it more acidic, which will ultimately induce the pain with several other chemical reactions. Now, the point of fermentation is to continually do this process to produce as much ATP as possible, because remember, glycolysis does not produce the majority of the ATP, okay? So in the presence of oxygen, the majority of the ATP is going to be made during oxidative phosphorylation, but oxygen has to be present, right? So in the absence of oxygen, or when the oxygen levels are very low, you're not going to be producing a whole lot of ATP, so you're going to run out of energy pretty quick. Now, let's compare fermentation with anaerobic and aerobic respiration. So we talked about aerobic respiration. This involves glycolysis, the citric acid cycle, the electron transport chain, and about 32 ATPs are made. And then again, the final electron acceptor is going to be oxygen. Remember, that's in the presence of oxygen. That's why it's called aerobic respiration. Now let's kind of compare both fermentation and anaerobic respiration. Now, anaerobic respiration is very similar to aerobic respiration, where glycolysis, the citric acid cycle, the electron transport chain. However, the amount of ATP can be as much as 32, sometimes more than 32, but as little as that fermentation produces. Okay, here the final electron acceptor can be a number of different things. It's just not oxygen. And that's why it's called anaerobic respiration. Now, fermentation, which is what we just discussed, yes, of course, it involves glycolysis, but it does not involve the citric acid cycle and the electron transport chain. And because glycolysis only produces 2 ATP, only 2 ATP will be made during one cycle of fermentation. And here, the final electron acceptor is going to be pyruvate in lactic acid fermentation and acetaldehyde in alcohol fermentation. Now, you can see the common thing among all of these is glycolysis, as I mentioned already. So you can see every single one of them utilize glycolysis. And remember, the glycolysis occurs in the cytoplasm. Okay, so fermentation will also occur in the cytoplasm in general, and then in the presence of oxygen, the pyruvate becomes oxidized. Right, so here in the absence of oxygen, there's other pathways in which the products of glycolysis will be dealt with. Okay, so it's not always it's not a mitochondrion that's dealing with this. So this kind of brings us to some terms which are used to describe the categories of organisms in relation to their ability to utilize oxygen. Now, we have what's called obligate anaerobes. Okay, so obligate anaerobes, obligate means obligated, so there it's required. Okay, so anaerobe means lack of oxygen, no oxygen, right? So uh, they are obligated to have no oxygen, so they're not going to be able to survive in the presence of oxygen. Yeast and many bacteria are called facultative anaerobes. So facultative anaerobes can utilize fermentation in the absence of oxygen or when oxygen levels are very low, and of course utilize the oxygen if the oxygen is present. Now, there's organisms that absolutely require oxygen, and we are one of those. So those are going to be, the term will be obligate aerobes rather than obligate anaerobes. Now let's look at this. I like to use this fork in the road kind of analogy, right? So we have this glucose, and it's being broken down by glycolysis. Remember, the products of glycolysis are 2 ATP, 2 NADH, and of course 2 pyruvate. Now, what happens to pyruvate, right? We don't know what happens to pyruvate unless we know whether oxygen is present or not, right? So if oxygen is present, pyruvates under aerobic respiration will enter the mitochondrion and then go through it, become oxidized, right? And then go through the citric acid cycle, and then the electron transport chain is where all the electrons which are extracted from that original glucose molecule will be dropped off in order to make even more ATP. Now, in the absence of oxygen, right? So in the absence of oxygen, we end up going to ethanol or lactate. Okay, so that's fermentation. That's basically what we just talked about. Again, this is presence of oxygen. So let's talk about the evolution, right, in terms of glycolysis and these processes. So glycolysis is a very old and ancient process, and the evidence of this is that basically every pathway utilizes it, right? So early prokaryotes they likely used just glycolysis to produce ATP before the oxygen started to accumulate in the atmosphere, right? So before like aerobic organisms existed or evolved, the oxygen levels were very low to minimal, none on Earth. So of course there's organisms that evolved that survived in the absence of oxygen. Now, um, glycolysis is also used in cellular respiration and fermentation, right? So it's a very widespread metabolic pathway, right? So again, this kind of exemplifies the fact that it is suspected to be the earliest and most ancient process because all organisms actually use it, and most of the pathways utilize it first in order to start to break down things during the remaining catabolic pathways. Now, it does occur in the cytosol, and that makes sense because earlier organisms did not have organelles, right? So before there were eukaryotes, remember the difference between eukaryotes and prokaryotes, right? Prokaryotes have no organelles, right? So they don't have, there was no endosymbiosis that evolved yet. It didn't happen yet. Okay, so again, glycolysis occurs in the cytosol. It does not need an organelle in order to produce ATP. Okay, so all of these signify the significance of the evolution of glycolysis. Now, let's talk about the crosstalk that I was talking about. So, glycolysis and the citric acid cycle, they allow other pathways to be connected to it, okay, and connected with each other. And we're going to kind of here. So remember, we eat carbohydrates, right? So those will be broken down into simple sugars and then further broken down, right, into components and eventually ATP. Well, we also eat proteins and fats as well. And then, of course, when we eat food from other organisms, we're eating their DNA. So their DNA can be broken down and be reused, right? So all the components will be reused. We're going to talk about the proteins and the fats along with the carbohydrates. So remember, during cellular respiration, in terms of the breakdown of glucose, we go through glycolysis, and then we've got glyceraldehyde 3 phosphate. Remember that in in the first uh, pathway, right? So the energy investment phase, and then of course this enters the energy payoff phase, and then we have pyruvate as the product. Now, looking at proteins, right? So remember, proteins are made up of amino acids. So the proteins will be broken down into their amino acids, so they'll be hydrolyzed. And then the ammonia will be broken off, and then the remaining parts of amino acids can be converted to pyruvate. So since they're converted to pyruvate, that could enter the glycolysis at the end of glycolysis. And then of course pyruvate can become oxidized in the presence of oxygen and then enter the citric acid cycle. With fats, remember, fats are made out of glycerol. Remember, the backbone is glycerol, and then 3 fatty acids. Well, if you cleave all the fatty acids off of the fats, then you have glycerol. Glycerol can easily be converted to glyceraldehyde 3 phosphate and then enter this main aerobic respiration pathway. Now further, okay, so other things here. Other parts of the amino acids can be converted to acetyl-CoA, which will then directly enter the citric acid cycle. And then the fatty acids are broken down into acetyl-CoA. So remember, acetyl-CoA is a 2-carbon molecule, and a coenzyme A is attached to it. Well, fatty acids are generally made in even numbers so that they can be broken down into these 2-carbon acetate groups with coenzyme A attached to that. This is called beta oxidation. Now, the amino acids, parts of the amino acids can also enter the citric acid cycle. Okay, so basically adding all of these reactants into these various points of the pathway keeps the pathway going, right? So it drives it forward. Remember, adding more reactants is going to drive a chemical reaction to products. And then the same thing with the fats, right? So when you're adding all of these things into this pathway, then it's going to drive to produce more ATP and ultimately during oxidative phosphorylation. Okay, so you can see there's crosstalk. We've got components of the breakdown of fats and components of the breakdown of glucose, which can be input as reactants at several points during this pathway in order to make ATP. Now let's talk about the regulation of cellular respiration, and it's regulated by remember that feedback mechanism that we talked about before. So feedback regulation. So let's kind of define that real quick first. Remember, feedback regulation is basically where the product of a pathway can come back and inhibit one or more of the enzymes early on in the pathway to slow or stop that pathway. Because if that product is building up, then you don't need to make any more, right? So it's a waste of energy to make more of a molecule in which the cell does not need. All right, so remember, I kind of mentioned when we were talking about glycolysis that phosphofructokinase is a key enzyme in the regulation of this particular pathway. So this enzyme is present in the first major phase of glycolysis. Okay, so that would be the energy investment phase, and it's regulated or controlled by a number of different molecules. Okay, so we're going to talk about each one of those. Now, let's say that this pathway is running really good and a whole lot of ATP are being made. Okay, so now we've got lots of ATP. Well, too much ATP, well, can bind to phosphofructokinase, right? So ATP, when it increases in concentration, it can bind to phosphofructokinase and then inhibit the pathway. So it inhibits that enzyme. Okay, so ATP can regulate phosphofructokinase. Now, citrate is the same way, right? So when the citric acid cycle is running, right? So we've got continuous input here, and this pathway is running nicely. Now, when lots of ATP is being made, also lots of citrate can be being made. Okay, so citrate is another molecule that can inhibit phosphofructokinase. So these are all remember, regulatory sites on an enzyme. Okay, so these are sites where molecules can bind in and change the shape of the enzyme so that it either activates or inhibits it. Okay, so this is similar to allosteric regulation, and these act as non-competitive inhibitors. Now, on the other hand, right, so this is stopping or slowing the pathway when we have too much of the products. Well, what if the pathway is not going fast enough? Or what if you're not eating enough food so that this pathway starts to slow down? But you're using some energy. Okay. Now, if you're using up all the ATP that's present, remember it gets hydrolyzed. Okay, so it gets hydrolyzed to ADP and then hydrolyzed again to AMP, or basically to AMP, and then pyrophosphate is cleaved off, which is two phosphates, inorganic phosphates, and then they'll be reused in some way. That means you have high amounts of AMP. Okay, so when there's high amounts of AMP, that means you're running low on ATP. When there's high amounts of AMP, that can bind phosphofructokinase and activate that enzyme to get this pathway going so that you can make more ATP as needed. Okay, so you can see there are at least 3 ways in which phosphofructokinase, one enzyme, can be regulated just by the products of this pathway. All right, so this is the end of chapter 9, right? So I talked about fermentation. Remember, there's lactic acid fermentation, there's alcohol fermentation, and fermentation processes. Aerobic respiration and anaerobic respiration all use the common initial pathway, and that's glycolysis. And remember, we don't only eat carbohydrates, so, proteins and fats must be broken down, and those components will be entered in the pathway at different varying points. So it kind of crosses into that pathway in some way, and that's where I'm talking about crosstalk. And then, of course, all of these pathways must be regulated. You do not want them out of control. Otherwise, it would be very inefficient, and it could be bad. All right, so thank you for listening.